In this video, I want to talk about the dark reactions of photosynthesis, which is basically the Calvin cycle, which is involved in carbon fixation. So where does this part of photosynthesis actually occur? This occurs, if you recall, in the stroma of the chloroplasts. So what we're doing is we're taking carbon dioxide and fixing it, right? We're fixing it into making glucose, essentially. So we said in a previous video that although that the dark reaction is light independent, the dark reaction can occur in the dark. So even though it's light independent, it does depend on the light reaction to a certain extent. Why is that? The reason why is because it uses ATP, right, and NADPH, NADPH from the light reaction. So the light reaction creates NADPH and ATP that the dark reaction can use. So during the day, a plant will go through the light reaction, create a bunch of ATP and NADPH, and at night, when there's no light, the dark reaction can occur. So the dark reaction can occur during the day as well, but at night, the light reaction can't occur, only dark reaction can. So if you had a plant, though, in the dark all the time, would it be able to survive? The answer is no, because it would need a source of ATP and NADPH. However, if it was in the dark and you somehow found a way to supply with ATP and NADPH without having the light around, then the plant could exist in the dark, and it could live in the dark. Okay, so the overall reaction of the Calvin cycle, we start off with 6 carbon dioxides, and we're going to invest 12 NADPHs and 18 ATP, and then we're going to get out of this. We're going to have 12 of those and 18 of those. 18 of those. Oops. Okay. So now, what does the actual Calvin cycle look like? Well, the point of it is to make glucose. Okay. There are 3 stages. The first stage being carbon fixation, the second stage being twofold, a phosphorylation step and a reduction step, and then the third and final stage is the regeneration of the 5-carbon acceptor. So what I want you to keep in mind, in order to understand this as best you can, I've written out this key here. So in orange, I'll be putting little numbers that indicate the number of however many molecules there are of that particular molecule. Anything in green will indicate the number of carbons in that molecule, and the number of carbons in purple that are boxed, that will be the total number of carbons present, okay, which will basically be this number times this number. Okay, so what are we starting off with? We're starting off with this thing called ribulose 1,5-bisphosphate, and we're starting off with 6 of them. Ribulose, you might recall, sounds a lot like ribose. Now, ribose had 5 carbons. It was a 5-carbon sugar. Ribulose is also a 5-carbon sugar. So we have ribulose 1,5-bisphosphate. So this thing has 2 phosphates on it, and it's 5 carbons long. Okay? So if we have 6 of them, we have 6 ribulose 1,5-bisphosphates, and each one is 5 carbons long. How many total carbons do we have? We have 30. So the reason I'm doing this is because it's very easy to get lost in this if you don't keep track of your carbons. Okay, so at this stage we have 6 ribulose 1,5-bisphosphate that in each one has 5 carbons, so we have a total of 30 carbons. So this first stage is the carbon fixation stage. So in this stage, what we're going to do is we're going to add carbon dioxides. We want to add 1 carbon dioxide to each one of these ribulose 1,5-bisphosphates. So we're going to have to add 6 of them. Okay, and each carbon dioxide is, of course, just 1 carbon. So that's a total there of 6 carbons, right? So if we're adding 6 carbons here, then we should have 36 carbons total in the next step. So if we're having these 6 carbon dioxides, each one is being added to a ribulose 1,5-bisphosphate, we would create 6 carbon molecules for a total of 36 carbons. But that 6-carbon molecule is very, very unstable, and it actually splits up into 3 carbon molecules, okay, which is 3-phosphoglycerate. So 3-phosphoglycerate, you've probably already seen it before. You should have seen it already in glycolysis and gluconeogenesis. It's a 3-carbon molecule with 1 phosphate group. So if this is a 3-carbon molecule and we have 36 carbons at this point, how many of them do we have? We have 36 carbons. So we should have 12 of these 3-phosphoglycerates. Okay, so we put these two together, the carbon fixation stage, and we made 12 3-phosphoglycerates. Once we have 3-phosphoglycerates, we're going to turn them into 1 3-bisphosphoglycerate. But what was the enzyme that created 3-phosphoglycerate from these two things here? This enzyme is called rubisco. Rubisco is basically ribulose 1,5-bisphosphate carboxylase oxygenase. Okay, so that's the enzyme that catalyzes this carbon fixation stage. So now once we have this 3-phosphoglycerate, this 3-carbon molecule, there's 12 of them, 36 total carbons. What we're going to do is we want to turn it into 1 3-bisphosphoglycerate. Now 1 3-bisphosphoglycerate, again, we've seen this in glycolysis and gluconeogenesis. This molecule has 3 carbons as well, and you should already be familiar with its structure. But now it's a bisphosphoglycerate. These are 3-phosphoglycerates. There's just 1 phosphate on each one of these things. Now we have 2 phosphates. So we must have added a phosphate for every single one. Since we didn't change, we still have 36 carbons. And since we didn't change the number of carbons, we should still have 12 of these, right? 12 times 3 will equal 36. But now each one of these things must have added a phosphate, right? That's the phosphorylation step of the second stage. So where do those phosphates come from? They come from ATP. How many ATPs did we use up? Well, we needed to add 1 ATP for each one of these 1 3-bisphosphoglycerates. So we must have added 12 ATP. So after we've added those 12 ATP, we have now 12 1 3-bisphosphoglycerates. So once we have this 1 3-bisphosphoglycerate, we want to turn it into glyceraldehyde 3-phosphate. You might recall that this step and this step, in this order, are from gluconeogenesis. So now, going from here to here, what would normally happen, this would be, first of all, it's going to be the reductions, the second part of the stage. So we would need a reducing agent. In gluconeogenesis, it was NADH, but in this case, we're actually going to use NADPH. So we are actually going to add 12 NADPHs here, right? 1 for each one of these 1 3-bisphosphoglycerates, and we're going to end up getting 12 NADP pluses. And of course, we're going to get 12 glyceraldehyde 3-phosphates. And the phosphates from these bisphosphoglycerates, 12 of them will hop off as inorganic phosphates. So what are we at now? At the second stage of the cycle, we phosphorylated and reduced the sugars, and now we have glyceraldehyde 3-phosphate, which is a 3-carbon molecule. We have 12 of them, so that's 36 total carbons still. So now what happens here? At this point, 2 of these glyceraldehyde 3-phosphates are going to leave, and 10 of them will continue on in the cycle. Now again, of course, each one of them is still just 3 carbons long, but now how many carbons stay here? Well, if there's 10 of them, if there's 10 G3Ps and each one is 3 carbons long, then we have 30 carbons staying in the cycle, and then that must mean 6 of them left. 2 G3Ps, 2 3-carbon molecules, 6 carbons. What do these 2 G3Ps do? These make glucose. Right? They leave to make glucose via gluconeogenesis. Right? One of these G3Ps will be converted into dihydroxyacetone phosphate, and then we'll go all the way back to glucose. So these 30 carbons here, these 10 G3Ps, what they're going to do is they're going to go back and become 6 ribulose 1,5-bisphosphate. Does that make sense? Well, if we have 30 carbons here, 30 carbons here, we don't need to add or remove any carbons. All we need to do is make sure that we add phosphates groups, right? Because we're going to have these glycerol 3 phosphates only have one phosphate. These ribulose 1,5 bisphosphates have two. So we need to add one phosphate for each one that we create. If we're creating 6 of them, we need to add 6 ATPs. Right? So this is the third and final stage, which is the regeneration of the 5-carbon acceptor. The 5-carbon acceptor, of course, is ribulose 1,5 bisphosphate. So then once we create these, once we've regenerated the 5-carbon acceptor, we can begin another cycle of the Calvin cycle. So I hope that was helpful, and thanks for watching.